Hi everyone, I'm Hannah. I am a PhD student in the Joy Lab and my marine science subfield is biogeochemistry. I didn't always want to study marine science. In undergrad, I was interested in plants. I went to Reed College in Portland, Oregon. I grew up in Georgia, but I went to this tiny liberal arts college on the other side of the country. So yeah, I studied these cycads and their microbial symbiont. I also spent a lot of time working at the nuclear reactor on campus, our little research reactor, which you can see here glowing bright blue from Cherenkov radiation. After undergrad, I wanted to take a break from the academic lifestyle and worked on a couple of tall ships. This one here is the schooner Zodiac, and we're in Puget Sound. And I realized I loved being on the ocean all the time. And I wanted to study science, and combining those two things seemed like a pretty obvious thing to do, and to do marine science. And the more I thought about it, the more sense it made. And I realized that I had actually always loved marine science and just had somehow thought that I wasn't cool enough to study marine science, like it was too sexy of a field for me. Um, but I had these very strong memories of a fifth grade class trip to the 4-H Center on Jekyll Island. If you're not familiar with the Georgia coast, it's got this whole series of barrier islands, some of which are really developed, expensive places, and some of which are nature preserves. Jekyll Island's a little bit of both. But we explored the marsh, we learned about the sand dunes, and I just remember all of it so clearly. And in my undergrad, I did an REU. This is Georgia Tech's Aquatic Chemical Ecology REU. This is my REU crew. In the background is actually the Sapelo Island Lighthouse. So we are at the University of Georgia Marine Institute on Sapelo Island in this picture. And going there and exploring the marsh again, I remembered all those things from fifth grade. And so these were two very formative experiences for me. And I realized how much I had loved them, and all my small marine science experiences in between, like tide pooling on the coast of California and things like that. So it took me a while to figure out what I wanted to do, but I made it here eventually. Now, what do I actually do? I study methane. Methane is a very important greenhouse gas. You can see in this plot here, here's CO2, our most important greenhouse gas in terms of anthropogenic radiative forcing. And right below it, we have a mix of other greenhouse gases. And of these other greenhouse gases, methane is the most significant. These two plots on the right, I know the top one is partly covered by my face, just shows you methane concentrations and how they've changed historically. You can compare those to CO2 concentrations, which we talk about much more. And then, of course, the projections for how those concentrations might change in the future under different climate modeling scenarios. So what does this have to do with the ocean? The ocean is actually home to a very large methane stock. 2.4 million teragrams of methane are in the sediments here. And this methane mostly stays there. Some of it is consumed by anaerobic methanotrophic archaea. Some of it, the 16 to 3,200 teragrams per year, does make it into the water column. I know this is a wide range. These things are really hard to constrain. Of that, not that much makes it into the atmosphere. And it's generally consumed by this aerobic microbial methane oxidation. And if you look here, you can see that measured oxidation rates range from 10 to the minus fifth to 10 to the third nanomoles of methane per liter per day. So this is many orders of magnitude, a really wildly variable process. And why it's so variable, what causes these rates to change so much, is something that interests me a great deal. So here's a little problem I've been working on lately. Comparing some different methane plumes in the Gulf of California. We have one cold seep, the Sonora Margin in the north here, and two different hydrothermal fields, the Guaymas Basin and the Pescadero Basin. So if we look at just the methane concentrations, we can see that our two hydrothermal sites, the Guaymas Basin here and the Pescadero Basin in gold, have the highest methane concentrations, with Guaymas being the highest. Note that this is on a log scale. However, when we look at the methane oxidation rate, we can see that the methane in Guaymas is turned over much faster than the methane in Pescadero. And this is of note because of how much more methane there is in Guaymas. This is like having a much bigger meal in Guaymas, but even though it's huge, they can eat it faster. So there are a lot of methanotropes working really fast in Guaymas. And we also see these different changes over depth of the vertical profile, with the turnover time getting shorter in Guaymas and longer in Pescadero. And what is driving these changes is something we're looking into right now. My little plug for Athens. I moved to Athens. I wasn't expecting to like Athens. I came to UGA in spite of Athens, not because of Athens. But I really came to love it. And yeah, the food's really good. There's a music scene. It's much harder to enjoy those things now than it was in the before times. But hopefully one day we will get to enjoy all these things again. And if you're around, maybe I will take you to some of my favorite places. Here's my email. Contact me if you like. Everyone have a great weekend. I hope you enjoy yourselves. I hope you learned a lot.